Recording. Good afternoon. Welcome to Thursday, March 26th lecture. We are going to do the same thing we did in the last one, and really the same thing we're going to do for the next almost 2 weeks. And that is, in lecture, we're going to talk about muscle physiology, and in lab, we're going to talk about the muscles. Our goal today is to, in the last lecture, we got through the first part of our muscle contraction, and that was communication at the neuromuscular junction. I know it was long, I know it was elaborate, but it is, like I said, the biggest chunk of the process. And so hopefully today we should be able to get through parts 2 and 3. That should also give us some time at the end to talk about the muscles. Again, we went through the muscles of the head. We didn't do everything that was on the list. Again, you are responsible for everything on the list, but we did hit all the ones that had origins and insertions and anything else that I thought might be a little tricky. But again, if there's anything I don't go over that you guys have questions on, make sure you ask that so that we understand that as well. Great question. So during the communication at the neuromuscular junction, what happens is the vesicle with the acetylcholine actually binds to the plasma membrane. So if you remember, basically that is what exocytosis is. Exocytosis is a vesicle, which is a piece of plasma membrane with some contents inside of it, coming to the surface of the plasma membrane, attaching to the plasma membrane, and as it attaches to the plasma membrane, it expels its material. So basically, if we cheat, we can draw this a little bit down here. So again, we have a vesicle. Here is the plasma membrane, and what happens is it comes down and attaches to it. So when it attaches to it, what happens is that it is first attached to the plasma membrane, and then once it attaches to the plasma membrane, what happens is it opens up the plasma membrane, and as it opens up the plasma membrane, its contents can be released. And that's essentially that process of exocytosis. So that's what it's binding to. It's binding to the plasma membrane of the synaptic end bulb, releasing the acetylcholine into the synaptic cleft. Great question. Any others? All right, so that is our game plan for today. Like I said, we did the muscles of the face. We are going to do at least many of the muscles of the trunk. We'll see how far we get if we get through all of them today. We don't have to get through all of them today, but we'll see what we can get through. Like I said, hopefully we can at least hit the important ones. And I guess before I started talking for 5 minutes, I should have, of course, asked, give me a quick show of hands to make sure everybody can hear me. Give me a thumbs up or a yes or a hand raise or something to let me know that you're hearing me okay. Perfect. Excellent. All right. So that's working. Great. Always good to know. The fact that you're asking questions and I'm answering them is always a good sign too. But again, I just want to make sure you guys can hear me, and that's awesome. Excellent. All right. Remember, there are 3 assignments as we talked about in the last class. I am going to have you turn in your unit reviews. Drop boxes have been set up in your excuse me, in your modules, same place where the Physio X1 is set up so that you can, oh no, plasma membrane is not the sarcoplasm, yes. No, it would not be the sarcoplasm because it's the plasma membrane of the synaptic end bulb. Remember, the plasma membrane of the muscle cell is the sarcoplasm. And actually, sarcoplasm is cytoplasm, it's the stuff on the inside, so you mean sarcolemma. So let's type that out because that was confusing for me. So let me say this again. The synaptic end bulb, we just call it the plasma membrane. And that is what the vesicles are binding to in the synaptic end bulb, just the plasma membrane in there. Sarcolemma is the fancy name we give to the plasma membrane of the muscle cell. So yeah, and sarcoplasm, of course, is the cytoplasm inside of the muscle cell. So those special words were just for the muscle cell. and this vesicle binding, the exocytosis that is occurring, occurs in the synaptic end bulb, so on the neuron side of things. Alrighty. So 3 more assignments, 3, not more, but 3 assignments you have to do. Your Unit 10 review is due on the Dropbox next week when we come back to class on Tuesday. And again, it's due by noon, just like it would be due on my desk by noon. 2 days later, your Unit 11 review is going to be due, again in the Dropbox, and then your PhysioX Exercise 2, which has 7 activities. So it's a lot, and I encourage you to be working on this while it's fresh, but I purposely have it due after spring break. Everything tells me that we are still having a spring break. Again, I've given you that extra time because I want to encourage you to work on it and make sure you understand all the physiology before we get to it as well. So that is the game plan, and that's what we're going to do today. So any questions on that before we get into the new material? All righty, then let's switch our view and take a look at our slides. Now, again, here, and this kind of answers the question that Oksana was asking. Notice here we have the vesicles that are contained with acetylcholine. They're coming and binding to the plasma membrane of our synaptic end bulb and releasing their contents by exocytosis. Here is the sarcolemma, the plasma membrane of our muscle cell, and in fact, here is the specialization. I know you can see that, but let's do that. Here is the the motor end plate on the muscle cell. This is where we have those invaginations, and this is where we have the channels, those specializations of the sarcolemma. And then all of this stuff we see on the inside would be our sarcoplasm, or the cytoplasm of our muscle cell. Now, we left off last class and we had talked about step 1. Can anyone remind me what step 1 is in our muscle contraction?
The decision to contract the muscle?
Yes, but what did we call it? What did we call step 1?
A neutral action potential?
Close, right, absolutely. That was the goal. But remember what we called it was communication at the neuromuscular junction. Right? That is step 1 in our process. That is what we talked about in depth in the last class. And as you guys already kind of pointed at, the goal of that, the ultimate goal, of course, is to get the muscle to contract. But in this case, the goal of this is to convert the neural action potential into a muscle action potential. Excellent. Lastly, what was the last step in that process of communication at the neuromuscular junction? Can you remember what the last thing we did when we were initiating it? All right, feeling shy today. All right. No, we'll get to the contractile cycle. You are right. That is the last step. There you go. What is going to happen is we are going in the last step. We, again, remember we got that depolarization to threshold. When we did that, we generated a muscle action potential, which I'm abbreviating it because I just wrote it above. And we did that at the motor end plate. And the muscle action potential spread down the sarcolemma. There you go. That was the last step of communication at the neuromuscular junction. And it's important to remember that last step. I guess I can see the confusion, the last step, because I said, so let's just change this to part 1. Part 1 is communication at the neuromuscular junction. The goal was to convert neural action potential into muscle action potential. And the last step of part 1 was depolarization at threshold, generate a muscle action potential at the motor end plate, and that muscle action potential spreads down the sarcolemma. I make a point of emphasizing this because, as we mentioned, this contraction of the muscle is a continuous process. So while this right here is the last step of communication in the neuromuscular junction, it is also going to be the first step of our second part of the process. And that second part of the process was excitation contraction coupling. All right, so let's take a look at that. Excitation contraction coupling. Last time we looked at the video associated with this at the end. I think this time it would be beneficial, because the drawing isn't quite as pretty, to take a look at the video first. So what I want to do is I want to go back to our, there we go, to the interwebs. This was the virtual anatomy lab. I was playing with that as we were getting set up. But here is our study area in the Mastering A&P. Again, the easiest way to find all the resources that you need are to go to the study by chapter. And we're still, well, today we're in muscle and muscle tissue for lecture. So we're going to click on that as well. And remember, we looked last time at the A&P flicks for the events of the neuromuscular junction. Now let's look at the A&P flicks for the excitation contraction coupling. Now again, what I want to tell you, whoops, hold on, pause that. Remind you before we do this is, again, some of the vocabulary will be slightly different. She's going to miss an important step near the end that we're going to make sure we emphasize. But I still think that the process is useful. And again, the visualization of this is really, really amazing. So let's go ahead and take a look at this. If I do that, do you guys get to still see it? Awesome. Let's see if that works.
Typically, a single motor neuron arising in the brain or...
Can you guys hear that? Can you hear the video when the video plays? Okay, perfect. All right, I'm gonna start it over since I talked over it.
Typically, a single motor neuron arising in the brain or spinal cord conducts action potentials that travel to hundreds of skeletal muscle fibers within a muscle. The sequence of events that converts action potentials in a muscle fiber to a contraction is known as excitation-contraction coupling. If we look at a single muscle fiber, we see that an action potential travels across the entire sarcolemma and is rapidly conducted into the interior of the muscle fiber by structures called transverse tubules. Transverse, or T-tubules, are regularly spaced enfoldings of the sarcolemma that branch extensively throughout the muscle fiber. At numerous junctions, the T-tubules make contact with a calcium-storing membranous network known as a Sarcoplasmic Reticulum, or SR. Where it abuts the T-tubule, the SR forms sac-like bulges called terminal cisternae. One portion of a T-tubule plus two adjacent terminal cisternae is known as a triad. The membranes of the T-tubule and terminal cisternae are linked by a series of proteins that control calcium release. As an action potential travels down the T-tubule, it causes a voltage-sensitive protein to change shape. This shape change opens a calcium release channel in the SR, allowing calcium ions to flood the sarcoplasm.
Notice right there, she did not use the term, but we can clearly see what that is. Notice there was a gate. That gate was moved by a voltage, and it opened a channel that allowed calcium to be released from the sarcoplasmic reticulum. That, of course, is our friend that we talked about before, the voltage-gated calcium channel. So there we had a voltage-gated calcium channel.
This rapid influx of calcium triggers a contraction of the skeletal muscle fiber. Thus, calcium ions are responsible for the coupling of excitation to the contraction of skeletal muscle fibers.
Hence the name, excitation, contraction, coupling. So close that. And now we can go back to the lecture. Perfect. Excellent. So notice here we have something similar. Where, again, we can see the anatomy. It's not quite as pretty as the three-dimensional view that we had before, but here we have the sarcolemma, the plasma membrane on the surface. And here we have those terminal cisternae that travel down into the, the transverse tubule that penetrate down deep into the center of the muscle cell. Here in blue, we have the Sarcoplasmic Reticulum, right? That specialized smooth endoplasmic reticulum that contains a large amount of calcium. And here we see the enlarged ends of it. The enlarged ends of it are those terminal cisterna. Two terminal cisternae and the transverse tubule collectively are known as a triad. And here we also have our voltage-gated calcium channels. All right, so any questions on that anatomy? We've seen it here in the pretty picture. We've seen it in the animation that is very pretty. Any questions on that? All right, now that we've seen it all pretty, let's have me draw it. Because now hopefully we have some understanding of what it is supposed to be. So we can go ahead and do this. And I'll cheat by using the straight line. So here is our sarcolemma. I'm going to, actually I should make this black. Or not. All right, there we go, try this again. There is our sarcolemma, and there's that here. And then here's our transverse tubule. There, excellent. We need to get our starting point. Our starting point, again, is going to be that invaginated motor end plate where our synaptic end bulb is communicating. This is where all of the steps, all the parts of step one are taking place. Here inside, we have, and let's draw it up here, our sarcoplasmic reticulum, and I'll put labels on all of these things in just a minute. Here is our terminal cisternae from one side. And again, remember, we're going to have a terminal cisternae from the other side. So we have those. And I'm going to just draw them as little gate-like structures. These are our voltage-gated calcium channels. And while there are a large number of them on the terminal cisternae, it is important to know that there are going to be some on the entire sarcoplasmic reticulum as well. All right. So far so good. So let's again, this is our, and I'm going to just abbreviate it here, synaptic end bulb. And this is our motor end plate, because they don't play a major role in the excitation contraction coupling. So we won't worry about having appropriate labels. This is our sarcoplasmic reticulum. And then the ends here, of course, are the terminal cisterna. Excellent. Move that a little bit there. Perfect. Let's remind ourselves that up here we have, and again, because this, actually, I think I have enough space to do that. Voltage gated calcium channel. Perfect. Channels, plural. Oops. Oh, sure. We have right here our our transverse tubule. Excellent. So we have that. So those are the things that your textbook or the animation just emphasized for us. But remember the other thing that we have going on, the thing that the sarcoplasmic reticulum and the transverse tubule wraps around, and I'll put it underneath it to emphasize that, is our myofibril. Oh, and now that I've done that, I need to undo that. All right, so this is our myofibril. And of course, our myofibril contains sarcomeres. So we need to draw one of those. We will start with our Z disc. And that's orbital Z disc, but you get the idea. And another Z disc. Excellent. We need our actin. And we need our myosin. And of course, our myosin has those myosin heads coming off of it. I'm just going to draw a couple. Excellent. All right, so let's label all these things. So again, we remember what they are. No, sorry, myosin. Apparently I can't type and think at the same time. Actin. Z disc. Whoops. So we have all of those things there. Perfect. However, there is one more thing we have not drawn, and I'm going to go ahead and draw this in purple to emphasize this. Oh, no, I didn't want to change that. All right, perfect, that's there. All right, so draw, line, purple, perfect. We have, we're not going to bother drawing the myomesin in there because right now it would just be in the way, but that is one of the structural proteins. Same thing with the titin. That's another structural protein. I'm not going to draw those because we don't need it. However, what we do need, what the calcium does that our animation did not remind us of, is what calcium actually does is moves those regulatory proteins. So we need to remember that there are two regulatory proteins. The first of those regulatory proteins is the tropomyosin. And I'll write it down here as a reminder. That tropomyosin, remember, is what is blocking the binding site so that the myosin head cannot grab onto the actin. But remember also, we have these handles on the tropomyosin that can move the tropomyosin out of the way. So that is the tropomyosin. And these handles are, remember, called troponin. Excellent. Now we have everything that we need to understand what is happening. Now, of course, what we are describing here, and I'm going to cheat and move this down out of my way. I know it's not in your way, but my annotation thing is in my way, so I'm going to move that out of the way so that I can write up here at the top. So remember, what we are doing now is part 2. And what is part 2 called again? I put you all to sleep with my drawing. All right, you guys are going to be all shy today. Makes it more boring for me, but all right.
Excitation, contraction, coupling.
There you go. Excitation, contraction, coupling. That is our part 2 that we are going to discuss. And again, remember, whenever we talk about one of these parts, we also want to talk about what our goal is going to be. And as the video, as we've already hinted at, our goal is going to be to use calcium to move the regulatory, there we go, proteins. Excellent. And as we just finished talking about before, we also know the starting point for this. So I'm going to write the starting point over here. We know the starting point. And again, as you see, I don't have a lot of room to write over here. So I am going to do a bit of abbreviating. Make sure you're not doing this on the exam, but I am going to do a bit of abbreviating. Our starting point is where we ended with communication at the neuromuscular junction. We produce a muscle action potential, and it spreads down the sarcolemma. All right, so up here, and let's go ahead and use blue. No, that's fine. I don't care. Up here, why won't you? There we go. We get this big, huge wave. This big, huge muscle action potential. It is, again, a big positive signal. And that big positive signal is going to spread down the sarcolemma. And then it is going to spread down the transverse tubule. So not only does it spread down the sarcolemma, but then it also spreads down. And since I already wrote it up there, I'll cheat and abbreviate it here, T-tubule. It spreads down the T-tubule as well. Now, when it's spreading down this T-tubule as a big positive signal, basically what it is going to do is it depolarizes the triad. Remember the triad, and let's go ahead and there and there. Remember, oops. The two terminal cisternae and the transverse tubules together, these things collectively form what we call our triad. And as we talked about here on our triad, when we depolarize those things, the key is we are going to reach threshold. And when we reach the threshold, we're going to open our voltage-gated calcium channels. And again, since I've written it out once, I will abbreviate it here, VG calcium channels. So the immediate effect of that muscle action potential spreading down the triad or transverse tubule is a depolarization of the triad. And when our triad depolarizes, it opens those voltage-gated calcium channels. And when we open those voltage-gated calcium channels, out of our sarcoplasmic reticulum, come on, we're going to get all this calcium that is released. Now, if we've learned anything, we know that calcium makes cells do wonky things. And cells don't want to do wonky things. So not surprisingly, our sarcoplasmic reticulum has a large number of calcium pumps. And the job of those calcium pumps are going to be to use ATP to move calcium back into the sarcoplasmic reticulum. This is kind of what we saw over in the synaptic end bulb, right? However, when these channels are open, much more calcium is going to come out than the sarcoplasmic reticulum can pump back in. So when the calcium is released, the net effect of that is that we get an increase in the calcium in the cytosol, in the watery component of our cell. Or you could say in the sarcoplasm as well if you wanted. Right? Those pumps are going to be trying to put it away, but more calcium is going to be coming out. All this massive amount of calcium is going to be coming out. All of these channels. And those poor little pumps can't keep up with it. All right. Now, notice as we talked about and as the picture shows, these sarcoplasmic reticulums sit right on top of the myofibril. So what ends up happening is that that calcium can come and actually bind to our troponin. So calcium binds to the troponin. And when that occurs, our troponin undergoes a conformational change. Which is a big fancy term, right? The kind of thing that you can impress grandma with, assuming you are, boarded up at home with her. But basically all it means is it changes its shape. Again, it's a big fancy word, but all it means is that troponin changes shape. And when it changes shape, what ends up happening is that when it changes shape, it rotates the tropomyosin out of the way. And so let's go ahead and cheat on this one over here and pretend it's been rotated. All right, here, we'll just do that because it's easier to get out of my way. There you go. So what ends up happening is, again, it rotates out of the way. And once it rotates out of the way, now our myosin is able to bind to the actin. And once myosin binds to the actin, our contractile cycle is going to begin. So I've run out of space down here at the bottom, so I'm going to finish my description over here. So again, calcium binds to the troponin, troponin undergoes a conformational change. It changes its shape. And our troponin rotates the tropomyosin off of the myosin binding site on the actin. So actin has that binding site for the actin, and it moves it out of the way. And once that occurs, we've reached our goal. Our regulatory protein is out of the way. And so now myosin can bind to the actin. And that is the end of the excitation contraction coupling. And guess what? That is going to be the very beginning of the contractile cycle, the third part of our muscle contraction. All right. Questions on that? All right, excellent. Perfect. Huh. All righty. No questions, no hands. Everything looks good. Excellent. We have looked at it here now on the whiteboard. Let's go ahead and let me move this now back up and out of my way up here. And then I can switch back to our lecture, and we can do the same thing here. We can see that exact same process here and how we go through it here. So again, we are now doing excitation contraction coupling, and because I want to emphasize it, let us make sure we understand that anytime we think of one of these steps, we want to think of the goal of this step, and our goal here is to use calcium to move the regulatory proteins. That process of excitation contraction coupling starts where communication at the neuromuscular junction ended. Our muscle action potential is produced at the motor end plate, and it travels down the sarcolemma. As it travels down the sarcolemma, it travels down our transverse tubule. Again, because our muscle action potential is a big positive signal, as it travels down the transverse tubule, it is going to depolarize our triad. Of course, if we're going to say that on an essay question, we have to define what the triad is. And that triad is the transverse tubule plus 2 enlargements of the sarcoplasmic reticulum. And these are the terminal cisternae. So 2 terminal cisternae and the transverse tubule makes our triad. And our specialization here is these voltage-gated calcium channels. So when that big positive signal comes down the triad and depolarizes our triad, it depolarizes our terminal cisterns. And when it does that, we are going to reach threshold. And when we reach threshold, again, there's that magic term. Let's write that as well. When we reach threshold, that is going to open those voltage-gated calcium channels. Calcium is going to rush out of the sarcoplasmic reticulum into the cytosol. Now, there are going to be pumps that are going to try to get it back into our sarcoplasmic reticulum, but there's more channels than there are pumps, and so more calcium comes out than can go back in. So we get an increase in the concentration of calcium in the cytosol. And here, in the illustration from your textbook, we see really beautifully how our terminal cisternae, how our transverse tubule, how the entire sarcoplasmic reticulum is wrapped around our myofibril. Here we have some myosin, here we have some actin, here we have the myomesin, so this is the M line. Way over here, we see a teeniest bit of the Z disc that we see there. So it is right on top of that myofibril. And that's important because when the calcium comes down to the myofibril, here we see part of our sarcomere in its relaxed state. Our myosin head wants to bind to the actin. Those black dots are the active sites, or we can think of them, and again, we can use them, call them either way. So let's write it up here. It's the active binding site, or we can call them what they are. They are the myosin binding sites. Active binding sites, myosin binding sites. But again, remember, they are on the actin. So they're not the actin binding site, they're the active, because they cause the active motion to do, or they're the myosin binding sites on the actin. But either way, the black spots are covered by that thread-like tropomyosin. And here is our troponin. However, in the presence of calcium, calcium binds to the troponin and causes the troponin to rotate. And as the troponin rotates, it moves the tropomyosin, and now those binding sites are open, and now myosin can grab. So it rotates the tropomyosin off the myosin binding site, and now myosin can grab onto the action, and our contractile cycle can begin. So moving those regulatory proteins is the end of step 2 and the beginning of step 3. All righty, questions on that? All righty. Let's take a couple more looks at this. Again, there are lots of illustrations that show this process. Again, here is one of the more simple ones. Here is our neuromuscular junction, where this acetylcholine is being released, opening up those cation channels, allowing the sodium to come in, reaching threshold, producing a muscle action potential. That muscle action potential spreads down the sarcolemma, down the transverse tubule, where it depolarizes our sarcoplasmic reticulum, releasing the calcium, which moves the regulatory proteins and allows the myosin and the actin to interact. But here is my favorite view. We keep looking at this from a side view, but notice what I love about this one is it's a cross-section view. With this cross-section view of our myofibril, here we see the actin. We know it is a dimer. It has 2 globular subunits. And here in yellow is the active site. This is what myosin wants to bind onto. Here is our thick filament, our myosin, another thick filament, that myosin, and one of the myosin heads. And these myosin heads are trying to grab onto the actin, but that pesky tropomyosin is in the way. However, there is a handle on the tropomyosin called troponin. And when our muscle action potential depolarizes our sarcoplasmic reticulum, those voltage-gated calcium channels open and calcium comes out. When calcium comes out, it binds to the troponin and causes the troponin to change its shape. And as it changes its shape, it rotates. And notice as it rotates, it moves the tropomyosin out of the way. Same thing's happening here. Calcium binds to the troponin, it changes its shape, and it rotates out of the way. And now that binding site is open, our myosin heads can grab, and our contractile cycle begins. All right. Questions on that? Excellent. So what we have done now, again, one more view of our initiation of, and I'm just going to abbreviate here, excitation, contraction, coupling. Here we have the cell at rest. We have our sarcolemma. We have our transverse tubules. We have our sarcoplasmic reticulum. And notice what's cool about this sarcoplasmic reticulum is it shows that calcium pump, because its job is to make sure that all the calcium is locked up inside of the sarcoplasmic reticulum. As we talked about when we just finished talking about the skeletal system, 99% of the calcium in your body is contained within the matrix of our bones, right? Our bone matrix is the primary location in the body where we have calcium. However, the sarcoplasmic reticulum of our skeletal muscle cells has the second highest density of calcium in the body. So the most calcium is in our bone matrix. The second most location is here in the sarcoplasmic reticulum. Massive amounts of calcium is kept in here. And we have these pumps to make sure that that calcium stays in there. With the calcium all locked away, right? And that's really the key, right? If you, I don't know how many of you have ever been to Folsom before or lived in Folsom maybe, but Folsom has a tremendous number of criminals in it, right? However, does that mean it's not safe to live in Folsom? No, of course it's safe to live in Folsom because the majority of those criminals are locked up in Folsom Prison. And as long as they're locked up in prison, they can't cause any problems. And that's what's happened here. All of our calcium is locked up in prison, can't cause any problems. Our tropomyosin covers our binding sites on our actin, and the muscle is relaxed. However, we generate that muscle action potential. The muscle action potential spreads down the sarcolemma, spreads down the transverse tubules, and it opens our voltage-gated calcium channels. And massive amounts of calcium come out. Now we have our Folsom Prison jailbreak. Yeah, we have the cops trying to put them back in jail, but way more coming out the front doors than can be pumped back in through the back. So we get a massive increase in calcium, and that calcium binds to the troponin, changes the shape of the troponin, moves the tropomyosin, and myosin can bind to the actin. All right. Questions on that? All right. So, as we've talked about, we need to understand how to initiate these processes, but we also need to know how to terminate them, how to turn them off. So we've initiated communication at the, we've initiated excitation, contraction, coupling. Now we need to terminate it. If you remember, when we terminated communication at the neuromuscular junction, what happened is we stopped producing muscle action potentials. Right? We stopped sending neural action potentials. We stopped releasing, we stopped letting calcium into the synaptic embole. We stopped releasing acetylcholine. We got rid of the acetylcholine. All those things we don't care about for this. Right now, we start here. If we want to terminate excitation, contraction, coupling, it starts with we stop producing, and let's do that. We stop producing muscle action potentials. The immediate effect of that is there's now no big positive signals spreading down the sarcolemma and spreading down the transverse tubules. So there's no more big positive signals spreading down the sarcolemma and down the transverse tubule. I'm going to abbreviate it, and down the T tubule. With no more, let's start over here. With no more big positive signal spreading down the T tubule, what happens is our triad repolarizes. Remember, repolarize was that term we meant that goes back to rest. Goes back to the resting membrane potential. Of course, if we do that, this cell is going to repolarize past threshold. And as we pass threshold, our voltage-gated calcium channels close. So no new calcium into the cytosol. All right, so no new calcium into the cytosol. However, just as we saw in, oh, I wonder if I can cheat. Let's do this. I like this. Oh, hold on. I want white. There we go. Perfect. Oh, I got rid of everything. All right. Now I have to go back. Oh no, I lost all of it. Ah, bummer. Oh well. So let's do this. Yes, sarcoplasm and cytosol in our muscle cell would be the same thing. All right, so bad news is I lost what I wrote before, but good news is that means we get to write it out again, and now we have a little bit more room. So let's talk about the second half of this, the second possible essay question here, and that is termination of excitation, contraction, coupling. Excellent. So this is our second possible essay question. Initiation, which we just finished talking about, and now termination. Again, it starts where we finished last time, and that is we stop producing muscle action potentials. All right. So that means there's no big positive signal spreading down the sarcolemma, oops, which means there's no big positive signal spreading down the T-tubule. Again, I'm abbreviating for space. That means that our triad is going to repolarize. And when our triad repolarizes, we are, and again, remember, this is where I wanted to do that. Repolarize means to go back to the resting membrane potential. When the triad repolarizes, our voltage-gated calcium channels close. So there's no new calcium into the cytosol, or again, sarcoplasm. Now, again, remember, cytosol is the fluid component. Sarcoplasm would include the organelles, but it still includes the cytosol. So cytosol and sarcoplasm are not completely interchangeable words, but it would be okay to use either of them here. So cytosol is the fluid component, sarcoplasm is the fluid component and the organelles. But the point is either way. So there's no new calcium that is there being released from the sarcoplasmic reticulum into the cytosol. However, we still have a lot of calcium in the cytosol. And this is where those calcium pumps are finally going to be able to make some headway. Our calcium pumps are able to remove the leftover or the remaining calcium from the cytosol with no calcium to bind to the troponin. The troponin goes back to its original shape. And when it goes back to its original shape, The troponin rotates the tropomyosin back over the active site on the actin, and our myosin can no longer bind. And there we have ended excitation contraction coupling. If you think about it, initiating was moving it out of the way. Termination is moving those regulatory proteins back. All right. So we have now initiated excitation contraction coupling. We have now terminated excitation contraction coupling. However, we did also talk about wanting to be able to modify as a third possible essay question on all of these processes. And what I will tell you is that when we talk about modifying this process, really what we're going to be talking about is the concept of fatigue. Right? When fatigue occurs, muscles don't perform as well as they would like. Now, there are a lot of reasons for this, and we don't fully understand them all. I can give you one simple example, though, and so we can see how this would work in this case. For instance, one of the things as muscles start to get tired is that they become more acidic. And as the cells become more acidic, as we know, our muscle cell is filled with proteins, and proteins are sensitive to pH changes. Yeah. And proteins are sensitive to pH changes. However, not all proteins are equally sensitive. As it turns out, one of the ones that is very sensitive is troponin. When the pH drops, troponin changes its shape, and it becomes harder for calcium to bind to it. So if you think about it, if we go through this process the way we've been doing, we generate the muscle action potential. The muscle action potential travels down the transverse tubule. It depolarizes our triad. It opens those voltage-gated calcium channels, and we dump a bucket of calcium onto our sarcomere. Now, even with a bucket of calcium, if it is harder for calcium to bind to the troponin because of the pH change, then fewer regulatory proteins are going to be able to move, fewer myosin heads are going to be able to grab and pull, and our muscle is going to get weaker. So fatigue is definitely a concept we will talk about, but it is a more complicated process and not something that we will talk about in this context. So what I'll tell you right now, when it comes to those quote unquote 9 possible essay questions, I will not be asking you how to modify excitation contraction coupling. So if you think about what we talked about in the last class and what we talked about today, fatigue can definitely be an essay question. When we get to that, we'll build up to that and talk about. But if you think about the essay questions from the last class, initiation of communication at the neuromuscular junction, termination of communication at the neuromuscular junction, modification of communication at the neuromuscular junction. There were 3 essay questions last time. So far, we only have 2 here. You could be responsible for describing how you initiate excitation contraction coupling, and you could be responsible for how you terminate excitation contraction coupling. So that is 5 possible essay questions. And in the next part of the lecture, we'll do 2 more. All righty. Questions on that? Excellent. We have 2 more essay questions under our belt. And again, Obviously, I want you to understand how this works and the process of it, but also knowing how to be successful with this on the exam is important. One of the things that I've talked to a couple students about, and I want to make sure to emphasize with you guys, especially because I'm taking the time to write this out. I'm taking the time to write this out here on the board, on the screen, because I want to make sure you're understanding the words that I'm saying. What I do not want you to do is just take a photocopy of that in your mind and regurgitate it back onto the page. Yes, you need to have all the details, but you also need to remember that when describing these processes, you need to describe them in your own words. If you just memorize word for word what I wrote down and then put that on the page, technically that's plagiarism, all right? Technically that's cheating. So make sure that you have this material, but also make sure that you are writing these things out in your own words. All right? Any questions on any of that? All right, excellent. This is a good natural stopping point. We are now 2/3 of the way through our muscle contraction. We need to get on to the second one. So let's go into the third part, contractile cycle. And for that, I will do that after the break. Let's go ahead and take a 10-minute break. So that means coming back at 1:15, and at 1:15 we will pick up from here. Oh, actually I lied. No, before we take a break, there is one more thing that I wanted to mention. As I mentioned, remember there is a massive amount of calcium that is kept here in our sarcoplasmic reticulum. Outside of the muscle cell, it is the highest concentration of calcium anywhere in the body. And one of the reasons it is able to keep as much calcium as it can inside the sarcoplasmic reticulum is because, and this is where I need to grab this, move that out of the way. There we go. Excellent. Cal's question. All the way out of the way. Perfect. Is that inside of our sarcoplasmic reticulum is a special protein called calsequestrin. Calsequestrin is a special protein that allows the sarcoplasmic reticulum to store and house a tremendous amount of calcium. As we've talked about, these alphabet soup terms, things that you almost certainly have to spell at some point or another, their name tells you everything about them. Cal refers to calcium, and sequestrin, to sequester, is what's happening to all of you guys, right? Back in ancient times, when people were on trial, there were things called juries. And if it was a very famous type of court case, they were concerned about the jurors getting information from the outside or learning things from the news. So what they would do is they would sequester the jury. They would lock them away at a hotel. They wouldn't get TV. They wouldn't get newspapers. They wouldn't be able to talk to people from the outside. It locks them away. And that's basically what this is. Calsequestrin sequesters or locks away the calcium. So it is a special protein found inside of our sarcoplasmic reticulum. All right, excellent. That now is everything that I wanted to talk about for excitation, contraction, coupling. So like I said, let's go ahead and take a 10-minute break. Now 10 minutes will be, we'll call it 1:16. So at 1:16, we will come back. So let me go ahead and write this here. Back at 1:16. And I'll remind myself to start recording. Excellent. See you in 10 minutes. All righty, we are back. Let's go ahead and clear all of that. All right. Next, our goal is to talk about step 3. And step 3, as I mentioned, is the contractile cycle, or what is also known as the sliding filament theory. Excellent. And like last time, we always want to talk about our goal first. So our goal for this is, like we said, we want to change the shape of the muscle cell, and that is going to produce a force. Or again, we can think of it as it's going to generate tension, right? Because tension is the term we use for the force that is generated. So that is our goal. So again, we can go over this on the whiteboard. We can go over this with the pretty pictures from textbook, and we'll go over this with the animation that we have as well. However, before we get to the animation, I think doing the animation first is useful. Give me a quick show of hands. Give me a yes if you thought watching the video first helped, or give me a no if you think watching the video after was better. All right, the few responses I got looks like the most wanted it first, so we'll go ahead and do that. Oh, well, now I have more afters, so now it's about even. All right, tell you what, then let's do it, so we'll mix it up. We'll do it both ways. We'll do it over, but I want to actually, this is one that I actually kind of like to go over first better myself as well. So let's do this. Before we even do either of those two things, there's a little bit of basic anatomy we want to go over, and I think I have that here. Excellent. Before we go over anything else and go through the process, we need to go back and remind ourselves of the anatomy of our sarcomere when it is at rest. Right? Again, as we've talked about, here is the Z discs, and those Z discs, oh, I'm off the screen, as you can see over here. My participation list is in the way. There we go, put that there. So here we see the Z disc over here. Here we see the Z disc over here. We have those. I do that. Perfect. All right, the two Z discs that we have there. We have our thick filament, the myosin here in the center, stabilized by the titin. Stabilized in the center by the myomesin. And remember, it's what defines our A band. Here we have our actin that has the binding site on it. It is bound to the Z disc, so it is anchored to and attached to the Z disc. And again, that area without the myosin is our I band. Here we have the center region with no overlap. This is the H zone. There's our M line. So we see all of that anatomy. That's all new. There's nothing new there. But the one thing I want to talk about are these myosin heads. Notice all of the myosin heads have a similar orientation. The myosins, and I'm going to exaggerate it on this side. Notice the myosin heads over here on the left project this way. And the myosin heads over here on the right project this way. So notice what we have here is the case of all of this. What happens is our myosin heads project away from the M line. So myosin heads over here on the left are projecting to the left. Myosin heads over here to the right are projecting to the right. So when a myosin head is primed and ready to do work, it points away from the M line. What I'm going to do in my drawing of this and explanation of it, and what the illustration is going to show as well, is to take just a small portion of that. So notice here we just take a small portion of this where we can see one single myosin, one single actin, and then the couple myosins. So we take one chunk here from the right side. So here is the thick filament of the myosin. Here's the thick filament from the myosin. Here are our myosin heads. And they are pointing away from the M line. So what we have here is them pointing away from the M line. Here we have our actin. Again, the yellow represents the binding site. And we can see that binding site is covered by the tropomyosin. And there are handles, the troponin. All right. So when we have this resting sarcomere, there's some basic important anatomy we need to know. The first is that the troponin covers the tropomyosin. The second thing we need to know is that these myosin heads, when they're at rest, are primed. Oops. right? And let's capitalize that, just emphasize it. Basically, what primed means is that they are ready to do work. There are two ways we can tell that they are ready to do work. The first is how they get ready to do work. The first thing they do is they split an ATP. They take an ATP and they rip off one of the phosphates. And of course, when you split an ATP, you get adenosine diphosphate and a phosphate. Now, normally, when we think of splitting ATP, we use that to do work. In this case, when our myosin head splits the ATP, it splits it in the ADP, it splits it into the phosphate. And of course, as a result of that, we get energy. But the key here is that the myosin head holds on to all of that. So notice they haven't given us anything to indicate the energy, but we have the ADP, we have the phosphate, it's holding the energy. It is ready to do work, right? But it can't do the work that it wants to do because what it wants to do is bind to the actin, and it can't. So it is ready to do work, and when it is ready to do work, we say that it is primed. All right? And notice, as we mentioned, when primed, the head points away from the M line. And again, it is primed, it is energized. And the other term we use for it is we say it is cocked, right? Like a gun. You cock back the trigger as you're getting ready to fire that gun, right? Or the little thing you cock back. So it's energized, it's cocked, it's primed. You can use all of those. It means the exact same thing. It is pointed away from the M line and it is ready to do work. All righty. Any questions on that? All right, if we have this, we understand our starting point, then what I can do is I can do the descriptions, I can do the drawings, we can go through this because I want to do that, and then we can go look at the pretty pictures of this again. So let's switch to the whiteboard. And let's go ahead and clear all the drawings from that. And again, here our goal is going to be step 3. That's 3. Contractile cycle. And again, our goal, change the shape of the muscle cell, right? Produce force, right? Generate tension. And let's do this. There. And there. Perfect. And I can get that and stick that out of the way. Excellent. That is our goal. Of course, we also know where we are going to start because the starting of contractile cycle is the end of excitation contraction coupling. And the start is that the troponin rotates the tropomyosin off of the active site on the actin. That was where we ended step 2, and that is going to be where we start step 3. Now, I'm going to do my pretty drawings like we did, and I'm going to do an even more simpler one than we just saw from the picture. So what we have here, let's not use red. Let's use black. So here is my actin. And then we need our Z disc. Oh, wait, hold on. That's going to be annoying to have to do it that way. So there's our Z disc. There is my myosin. And here, oh, you know what? Let's do this. I'm going to cheat and give myself a little more room so that this is not drawn to scale. But if I make it nice and big, then inside of my myosin, I can have my ATP and my phosphate. Perfect. So again, here I have a primed myosin head with the ATP and the phosphate on it. And it's ready to bind to the actin. And all we need for that to occur is for us to be able to rotate, right, that tropomyosin out of the way. So if we're able to rotate the tropomyosin, out of the way, then our binding site is exposed and myosin can finally start to do some work. So again, this is our starting point. Any questions on that? All right, excellent. Complete quiet tells me that you guys completely understand this. All right, now there's one other key to this part as well, and that is this phrase over here. The cycle, remember, is a continuous process. So this is something that is going to go around and around and around and around. So let's talk about those steps and let's see what happens first. We got to quickly do our drawing again. So there again is my actin. I'm not going to bother labeling it all this time because at this point we should know what these things are. The first thing that primed myosin head is going to do is to come up here and grab onto the actin. So when that tropomyosin is out of the way, myosin grabs the actin. Of course, saying myosin grabs the actin makes a tremendous amount of sense. It is what is happening after all. However, as we know, our anatomists love to name everything. So the myosin binding to the actin is given a fancy name. Basically, it says that it forms a cross bridge. All right, that is the fancy term. But basically, all that means is that the myosin head binds onto the actin. I don't know why I capitalized that, but there you go. Binds to the actin. That is the first thing that happens. Now, of course, I still need to emphasize that in here, I've got my ATP and my phosphate. Once that myosin head can bind to the actin, now it is finally able to release its energy. And when the myosin head releases its energy, and let me write this down here as I talk about it. Oops, no, don't want to do that. Put that back. Once myosin binds, it can finally release its energy. Remember, our myosin head is a motor. It uses chemical energy to produce a mechanical energy. a force. And that's what it's going to do. It releases its energy, and again, big fancy word, it is going to perform a power stroke. All right? That is what our myosin head is going to do. It's finally going to be able to release its energy, and it does that by performing a power stroke. What that means is basically the myosin head pivots towards the M line. Oops. So if we draw our drawing one more time, there is our actin. And let me move that up a little bit. Come on. There we go. Perfect. There it works. And then we draw our Z disc. And we draw our myosin. What our myosin head is going to do is it is going to pull on the actin, and it is going to pivot. So what happens is that our myosin head pivots going forward this way. And as it goes forward this way, if you think about what's going to happen, it's going to pull on the actin. And since the actin is connected to the Z disc, it is going to pull on the Z disc. So when it performs this power stroke, myosin pivots towards the end line. Oh, I wrote that already. And as it pulls on the actin, which pulls on the Z disc. Now, remember, this is happening on one side of the sarcomere. The same thing is happening on the other side as well. So if you think about what's happening when this is pulling on us, this basically, and I'm going to steal a space up here to write on it, what happens from the two sides is that the two Z discs are pulled towards each other. All right, hopefully that makes some semblance of sense. As they are pulled towards each other, if you think about it, what that means is that our sarcomere gets shorter. If our sarcomere gets shorter, then that means our entire myofibril gets shorter. Because not only is this sarcomere one of the units of our myofibril, but it's happening in all the sarcomeres, so they're all pulling at the same time. Our myofibril gets shorter. Our myofibril, remember, thanks to those structural proteins, are connected to the muscle cell, so our muscle cell gets shorter. And as we've seen, our muscle cell has an endomysium. It's bundled into a fascicle by a perimysium, and that perimysium is connected to the muscle by the epimysium. So if a muscle cell gets shorter, then the muscle gets shorter. And as that muscle gets shorter, it pulls on the bones, and we generate a force. This is how we get, our tension. All right. We produce our tension, we generate our force, and that happens right here. This power stroke is how we are producing our force, how we're changing the shape of the muscle cell, and how we are generating that force. All right. Now, one more thing happens during this power stroke. During the power stroke, and there's a little bit more involved in it than this, but we're going to simplify it a teeny bit. During the power stroke, the other thing that happens is that the ATP and the phosphate are released, right? Oops, that doesn't work there. So there you go. Put it that way. And oh, let me grab that. That's what I want. There we go. So there you go. Our ATP and our phosphate are released. So we are using the energy. We perform that power stroke. All right, we're going to pivot. And during that process, the ATP and the phosphate are released. Excellent. There you go. We have accomplished our goal. Oh, yes. Thank you. I'm sorry. But I have to, I don't think I can, there's no way to get back in there. So delete that. Thank you. I will write that again. ADP. Thank you for catching that. And the phosphate are released. Thank you. All right, perfect. I told you, I'm not typically the kind of person who can walk and chew bubblegum at the same time. So this having to type and draw and write all at the same time, it's very, very confusing for me. It's very exhausting. I take a nap after every lecture. I'll be happy when these are all recorded. And if we have to do this next year, I can just play them and sleep. It's going to be awesome. All righty. Excellent. This has been our goal. This was our goal. Our goal was to produce our tension. And here, 2 steps into the process, we have done that. And that is awesome. Except 2 things. One is that one myosin head grabbing and pulling is going to generate some tension, but it's not going to produce enough. And definitely not enough for us to be able to move our body entirely through space. The other issue we have is this is a cycle. It has to continue. I have to continue this cycle around. So what has to happen now is we need to continue this process. And here's the other problem. Our myosin head has used its energy, had released the ADP and phosphate, and now it's stuck here. So the other thing we need to do is we need to get the myosin head to let go so that it can be primed again and ready to do work. And the way we do that, the nice thing about the ADP and the phosphate leaving is now there is space for a new ATP to bind. And that is exactly what happens next. So we have to draw one more of these portions of a sarcomere. There's my actin. Here is my Z disc. Here is my myosin. And here is my myosin head. Great question. So the question is, oh, everyone see it. The myosin head never get damaged? No, absolutely they get damaged, right? And again, you knew that because if you're stuck at home, you're doing a lot more exercise. You can't get to the gym, so you do 75 push-ups today. You do 75 push-ups today, how are your arms feeling tomorrow? They feeling good? Happy and you're ready to do 75 more push-ups tomorrow? No, probably not. Those muscles are going to be sore. And the reason muscles are sore, part of the reason muscles are sore is that, yeah, this generation of force is damaging. So yeah, absolutely, we do damage these proteins. But remember, we're looking at one myosin head. A single myosin filament has thousands of myosin heads on it. A single muscle cell has thousands of myosin filaments inside of it. So we're talking about each muscle cell having millions of myosin heads that grab and pull. So if a good 100,000 of them get damaged, you still have plenty more. So yes, during this process, proteins can be broken, proteins can be misaligned. That's actually one of the things that leads to fatigue. But in general, in less damage, these myosin heads can continue this process. Now, what we need our myosin head to do is we need our myosin head to break the cross bridge. Right? Or essentially, we need it to let go of the actin. It turns out the way we do that, and again, let's draw my arrow. So we want that myosin head to let go. And the way we do that is with a new, and I'm going to change the color to emphasize this. A brand spanking new ATP is going to come and bind to our myosin head. So what happens is a new ATP binds to the myosin head. Period. That is it. The ATP binds to the myosin head, and just the act of the ATP coming in and binding to it is enough. Oops. Do that. To get the myosin head to let go. Notice it is not using the energy of the ATP. Just merely the act of the ATP binding to it causes the causes the myosin head to let go. ATP, again, great question. Where does the ATP come from? Remember, inside of all your cells, all of your pumps, all of these other things are going on. Your cell is constantly in need of ATP, the way your cell constantly needs glucose. So there's a ton of glucose floating around in the cytosol. There's a ton of ATP floating around in the cytosol. But you have hit on a very important point. When we come to contracting a muscle and we want to sustain that contraction, are there going to be limits to how much ATP we have floating around inside of the cell? Absolutely. And so if we're going to sustain a contraction, do we need to be able to make new ATP? Absolutely. And so that is actually, once we understand this mechanism, where we get that energy, where this ATP comes from, is actually going to be one of the really important questions we're going to need to ask and answer. But for right now, this is just ATP that is floating around from the cytosol. So a new ATP that is floating around in the cytosol binds the myosin head and causes the myosin head to let go. Notice it's not holding on to the actin anymore. So that's good. Now the actin is exposed and something else can grab onto that handle if we need it. But notice our myosin head is not ready to do work yet. To do work, it has to be revitalized. And the way it revitalizes, to split the ATP. When it splits that ATP, right, then what happens is twofold. We split the ATP into ADP plus the phosphate, and that splitting process energizes the myosin head, and it primes it to point away from the end line. And now I got to find a space to sneak that in. Here, we'll put this here for right now, and then move that up there, move that up there, and then let's just put this right there. Excellent. So notice this whole thing is a continuous cycle. So it splits that ATP. When it splits that ATP, that primes it, cocks it, points it away from the M line. It holds on the ATP and phosphate, and assuming the tropomyosin is still out of the way, that process can continue. It's going to bind and form a cross bridge. When it forms a cross bridge, it is going to perform a power stroke. When it performs a power stroke, it is going to use its energy, releasing the ADP and phosphate, and it's stuck there until a new ATP comes in, binds to it. When the ATP binds to it, it breaks the cross bridge. And once it breaks the cross bridge, then that splits the ATP, revitalizing the myosin head so that it can grab and pull again. And that is the contractile cycle. Right? And being a cycle, we want to emphasize the part that it does continue. Right. Questions on that? All righty. So that is the big elaborate process of the contractile cycle. Now that we've gone through it with my pretty pictures, let's go through it. And hold on, whoops, cancel that.
I have a question.
Yes.
So the ADP and the phosphate form only when the myosin head splits the ATP?
Ask that again.
So ADP and the phosphate get formed only when the myosin head splits the ATP?
Yeah, remember here, I'll cheat. If you remember back in ancient times when we talked about chemistry, we talked about what ATP was. ATP was adenosine diphosphate. Basically, it was a 5-carbon sugar. And if you remember, it was connected to a nitrogen base. And then off of that are 3 phosphates, 1, 2, and 3 phosphates. Remember, these phosphates are really, really negative things. So to get those negative things to come together requires a tremendous amount of energy. So it forms a massive amount of energy to form this bond to make adenosine monophosphate, adenosine diphosphate. And then to add a third on requires a major, major amount of energy. So we have this one ATP molecule, and what your myosin head does is it basically grabs it and splits it off. It breaks it off. When it breaks that bond, it releases all of that energy. So all of the energy is released when it breaks that bond. And that's the energy that this is going to use to do the work. Right? So it's got all this energy bound out, and that energy is what it's going to use for that power stroke. And notice what it's left with holding on to is adenosine diphosphate and the phosphate. So when it splits it, normally, like for instance, when we think of a calcium pump, basically the ATP comes to it, it phosphorylates, which is the fancy word for saying that it rips off a phosphate and releases that energy. It uses that energy to pump calcium where we want it to go, and then the ATP and the phosphate go away. What's different about the myosin head is that it holds on to them. It splits it, but then it stores that energy. It holds on to all the pieces, and those pieces are what help it to bind and what help it to form the power stroke. When it binds and performs the power stroke, it uses the energy and it releases the ADP and phosphate. All right. Technically, it uses the phosphate to help to strengthen this bond so that when it pivots, it can pull on the actin. But you don't have to go into that much depth. Just know that it's storing the ATP and phosphate. It forms that cross bridge. And when it forms that cross bridge, it's finally able to use that energy that it's been storing. All right. So here again, when it's binding, it has the energy. But here, it finally gets to use that energy to perform that power stroke. And during that process of binding and forming that power stroke, the ADP and the phosphate are released. So now there's space for a new ATP to bind to. Does that help? I'll take silence as a yes.
Yes, sorry.
Okay. Excellent. All right. There's one other point. Again, I know you guys probably haven't thought of these things in this term, but this in particular right here is a concept that I know many of you are aware of, right? Because like I said, myosin head uses the energy to perform that power stroke, but then it's stuck. It can't let go. The only way it can let go is if an ATP binds to it. But that's okay because ATP is always present in the cell. Cells are always making ATP. There's always ATP in the cell. In fact, what would your condition be if you weren't making ATP? Yeah, exactly. Dead. You'd be dead. So think about this. You die, whatever position you're laying there, sitting there in your chair, listening to this lecture, you die, and about 12 hours come by, and one of your loved ones comes to check on you. And as they're checking on you, one of the things that they've noticed is your body is locked into whatever position you were in when you died. This is why that happens. What happens is when you die, all the ATP in your cells are depleted. They don't make any more ATP. And your myosin heads basically grab onto the actin and don't let go. What do we call that condition where the body's locked into whatever position it was in when you died? Anyone know? I know you know. Rigor mortis. Rigor mortis is that condition where the body is locked in the position that it dies, and that's because the myosin heads lock onto the actin. Notice when you die, you don't instantly go into rigor mortis. It takes some time. What happens is all the ATP gets used up, and then calcium starts sneaking into the cells. As there's calcium sneaking into the cells, we don't have any pumps to get rid of it anymore. So calcium can bind to the tropomyosin, to troponin, move the tropomyosin out of the way, and all our myosin heads are primed. So they grab and pull using their energy, and then they're locked in place. Right? And so it takes some time for that to occur. And then, 24 hours later, as the body starts to break down, as the proteins start to break down, that tension is lost. So the person doesn't stay in that rigid position forever, but the state of the body, right, whether they're in rigor mortis, whether they've passed out of rigor mortis or haven't gone into it yet, is one of the things that, criminologists use to determine how long someone's been dead. So again, anyone who's watched a CSI show probably has heard of rigor mortis before. This is what causes it. The myosin heads grabbing and pulling and not being able to let go. All righty. Any other questions on this?
Yeah, I have a question.
Yes.
So in the first and second drawing, it says ATP, and the phosphate, is it supposed to be ADP as well?
It is. I screwed it up in all of them. Thank you for catching that.
Perfect. Thank you.
Yeah, no, here. I don't know an easy way to fix that, so we'll just cheat and do this. There. And there. Thank you so much for catching that. So now ADP. ADP. Thank you so much for catching that. Excellent. All right. And one of the things that I noticed is there's a way to, where'd my whiteboard go? There it is. There is a save option where I can save these whiteboards. So I've saved them. I'm going to try to post them to see if that helps. We can look at these again. I'll see. I haven't looked at them yet to see if it's any good, but if they are, I will post them on the Canvas site. Thank you for catching that. Have any other errors? That one's ATP. Those are all ADP, phosphates, energy. Perfect. Excellent. Any other spelling errors or any other errors are on me. Thank you for catching that. All righty. And that might have been where some of the confusion before was coming from as well. So I do apologize for that. Excellent. Make that prettier. All right, perfect. So now that we've seen it here, let's go ahead back to the lecture and see it here. Here it's going to be nice and pretty. So again, this is our starting point. Annotation, highlight. Our myosin heads are primed. Notice it split the ADP and phosphate. It is pointing away from the M line, because remember, we can see we're on the right side. So the M line is over here. The Z disc is over here. But it's storing the energy. It can't use the energy yet because it can't bind to the actin, because the tropomyosin is in the way. All right. So our contractile cycle begins with the muscle relaxed, with where excitation contraction coupling ends, and that is the exposure of the active site. Calcium binds to the troponin. Troponin changes its shape and rotates the tropomyosin, and our active site is revealed. Again, this is the end of excitation contraction coupling, so it is the beginning of the contractile cycle. Once that occurs, now notice our primed or energized or cocked, whichever term you like, myosin head is able to bind to the actin. And again, remember, we give that a fancy name. We say that it forms a cross bridge. Right? And again, it's only these primed myosin heads that are able to form the cross bridge. So as long as they're pointing away from the M line, they're able to bind to the actin and form that cross bridge. Once they form that cross bridge, they are then going to perform their power stroke, which is a fancy way of saying it is going to pivot towards the M line. This is where we release our energy, right? This is where we are making the sarcomere shorter, which makes the myofibril shorter, which makes the muscle cell shorter, which makes the muscle shorter. This is what generates the tension. This is what is producing the force. These power strokes of our myosin heads, grabbing and pulling on the actin. And as we talked about during that power stroke, it is using the energy, but notice it released the ADP and phosphate. Once it releases the ATP and phosphate, a new ATP will bind to it. And notice the picture from your textbook doesn't show this. It will bind to it, and that breaks the cross bridge. But remember, it's not ready to do work yet until it splits that ATP into ADP and phosphate, and then it is revitalized and ready to go again. So again, we break the cross bridge first, then we split the ATP, that recocks or revitalizes the head, ready to form the new cross bridge, and the process continues. These are the pictures from your textbook, but I really like this picture, as you can tell, because I totally mimic the one that I do off of this illustration, where we can say this. Here is our starting point where we started, with that primed myosin head. Actin sites are exposed, so the myosin head grabs onto the actin. Notice it has split the ATP into ADP and phosphate and has the energy. Once it forms the cross bridge, it performs its power stroke, pulling on the actin, using the energy, performing that pivot towards the M line, and it releases the ADP and phosphate during that process. Notice a new ATP comes in and binds to it, and that breaks the cross bridge. But remember, it's not ready to do work yet. It first has to be primed again, pointing away from the M line, and to do that, it splits the ATP. When it splits the ATP, it primes it, and as long as the binding site is exposed, it can grab and it can pull again. And that's what this does. Every single one of these myosin heads grabs and pulls at a rate of about 5 times in a second. Right? If you think about it, it does what you would do if you were climbing a ladder. If you were standing at a base of a ladder and you wanted to climb it, here's what you would do. You would reach out and grab the rung. Once you grab the rung, you would pull down. Once you pulled down, you would let go, and then you would reach up to grab another rung, right? So reach, grab, pull, let go. Reach, grab, pull, let go, right? You can do it like I'm doing it right now with your hand, and imagine trying to do that 5 times in a second. That's how fast these myosin heads grab and pull. And again, each myosin head by itself doesn't do much. one contractile cycle of one myosin head only makes the muscle cell like 0.5% shorter than it was before. But like I said, we have thousands of these that are all working at the same time, and that is what generates our force. That is what shortens our sarcomere, which shortens our myofibril, which shortens our muscle cell, and so on and so forth. All right. So that is the contractile cycle. Now, again, we also, as we talked about, can talk about termination of this process. How do we stop this process? How do we stop this continuous cycle? Can anyone think of a way to stop this, or do arm muscles, once we start contracting a muscle, it stays contracted for forever? True, if you die, you definitely can't get this process to occur anymore. But again, flex your arm, lift up your mouse off of the table and then put it back down. When you lifted that mouse off the table, did your hands stay rigidly contracted like that for forever? Hopefully not. We need to get this to stop. There you go. Well, remember, we're not producing calcium, but we are releasing calcium. So you are right. If we put the calcium away, remember in our excitation contraction coupling, we put the calcium away. And if we put the calcium away, remember what happens is our troponin changes back to its original shape. And when it changes back to its original shape, there, spread that out there. Then the troponin rotates the tropomyosin over the active site. And if tropomyosin's over the active site, when our cycle comes around, myosin can't bind. And if myosin can't bind, the contraction stops. Just that simple. So simple, in fact, that it doesn't even really make a good essay question, does it? So termination of this, stopping of this, is really just rotating the tropomyosin back in the way. And so that's not likely, let me rephrase that, it will not be one of your essay questions. So again, if you think of our essay questions, and we'll write this after the break, but if you think about our essay questions, so far now we have 6, and we are going to add 1 more. So there will be a total of 7 possible essay questions from this process. And as I've mentioned, I guarantee 2 of them will be essay questions on the exam. All righty. All right, excellent. All right, now we've done it with my drawings. Now we've done it with the pretty pictures from your textbook. Let's do this one more time, and that is by looking at the illustrations, the animation from your study guide. So if we close that and go back here, here's our Crossbridge cycle.
The contraction of a skeletal muscle generates the force necessary to move the skeleton. A contraction is triggered by a series of molecular events known as the Crossbridge Cycle. In a skeletal muscle fiber, the functional unit of contraction is called the sarcomere. A sarcomere shortens when myosin heads in thick myofilaments form cross bridges with actin molecules in thin myofilaments. The formation of a cross bridge is initiated when calcium ions, released from the Sarcoplasmic Reticulum, bind to troponin. This binding causes troponin to change shape. Tropomyosin moves away from the myosin binding sites on actin, allowing the myosin head to bind actin and form a crossbridge. Also note that the myosin head must be activated before a crossbridge cycle can begin. This occurs when ATP binds to the myosin head and is hydrolyzed to ADP.
Notice what just happened there. When the ATP bound to it, notice the myosin head let go of the actin. Again, remember, just myosin binding to ATP is what breaks the crossbridge. She doesn't emphasize that, but she showed it to us really nicely. So let me move this out of the way so I can get back. There it is, perfect. Here, and then I can go back a little bit so we can watch that again. Oh, no, I didn't want to do that.
Myosin heads in thick myofilament crossbridge is initiated when calcium ions, released from the Sarcoplasmic Reticulum, bind to troponin. This binding causes troponin to change shape. Tropomyosin moves away from the myosin binding sites on actin, allowing the myosin head to bind actin and form a crossbridge. Also note that the myosin head must be activated before a crossbridge cycle can begin. This occurs when ATP binds to the myosin head and is hydrolyzed to ADP and inorganic phosphate. The energy liberated from the hydrolysis of ATP activates the myosin head, forcing it into the cocked position.
So notice there she showed us two things. What she showed us is the breaking of the crossbridge, which is the ATP binding to it and the myosin head letting go. But then we saw how it then split the ATP into ADP and phosphate. And as it did that, that is what primed it. So the priming occurs when you split. Just the binding of the ATP got it to let go. So there's actually two steps there. ATP binds and it breaks the crossbridge. Then it splits the ATP to become primed. And that's what we have here.
A crossbridge cycle may be divided into 4 steps. Step 1, crossbridge formation. The activated myosin head binds to actin, forming a crossbridge. Inorganic phosphate is released, and the bond between myosin and actin becomes stronger. Step 2, the power stroke. ADP is released, and the activated myosin head pivots, sliding the thin myofilament toward the center of the sarcomere.
Now again, notice she showed separately how the ATP and the phosphate are released and the significance of that. We're clumping it all together into one process. We just know that to form that power stroke, it has to release the ATP and phosphate. Basically what happens is it grabs hold stronger and then it's able to release the energy. But we don't have to worry about separating that into 2 steps. So we'll just know that during the power stroke, it releases the ADP and phosphate.
Step 3, crossbridge detachment. When another ATP binds to the myosin head, the link between the myosin head and actin weakens, and the myosin head detaches. Step 4, reactivation of the myosin head. ATP is hydrolyzed to ADP and inorganic phosphate. The energy released during hydrolysis reactivates the myosin head, returning it to the cocked position. As long as the binding sites on actin remain exposed, the crossbridge cycle will repeat. And as the cycle repeats, the thin myofilaments are pulled toward each other and the sarcomere shortens. This shortening causes the whole muscle to contract. Crossbridge cycling ends when calcium ions are actively transported back into the sarcoplasmic reticulum. Troponin returns to its original shape, allowing tropomyosin to glide over and cover the myosin binding site on actin.
And there you go. Just that simply, we have contracted a muscle. We have gone all the way from the point where we made the decision all the way to the point where the muscle has contracted. It is generating a force and it is moving the body through space. It really is just that simple. All righty. Questions on that? All right, excellent. This is a good step. So our last thing that we need to talk about, let's do this. So we've done all of those things now. The last thing we need to talk about, as we mentioned, is to talk about a last step, right? As I've mentioned, when we think of terms of possible essay questions, and again, obviously our goal is to understand this information, but being the sophisticated students that you are, you also want to be thinking about how you're going to be tested on this material. So if we think of all the possible essay questions, all the processes we have described over the past 2 days, the first one is initiation of communication at the neuromuscular junction. And I guess it helps if I spell communication correctly. Excellent. Second was termination of communication at, and I'm going to abbreviate it here, the neuromuscular junction. And 3 was modification of communication at the neuromuscular junction. And of course, that was when we talked about things like Botox and things like that, etc. Right? Question 4 is initiation of excitation, contraction, coupling, termination of excitation, contraction, coupling, and now contractile cycle. Those are the 6 processes that we have talked about that could be possible essay questions, right? And those are our 3 steps: communication at the neuromuscular junction, excitation, contraction, coupling, and the contractile cycle. However, if you remember at the very beginning of this section, we did talk about that if there was a 4th step, that 4th step would be taking all of the terminations together and putting it together into relaxation of the muscle. And so one of the ways that we can make sure we understand this information is by putting it back together. We are artificially chopping it up into easily digestible pieces. But one of the things to think about is this is a continuous process. So what I'm going to do based on the time is let this sink in, and then when we come back, we will put all the pieces together of the termination of all of these processes to see if we can make sense of how we would actually relax the muscle, and know that that could be the possible 7th essay question. So as I mentioned, these are the 7 possible essay questions. Like I said, I guarantee there will at least be 2. And depending on the format of the exam, because it's looking more and more like we may have to continue this online, it may be even more. There may be a 3rd at least that comes from this. All right, and so let's think about this. Let this information sink in because we've talked about a lot, but then we have one more big important process to talk about. We'll put all the pieces together and talk about that 4th step, that quote unquote 4th step, right? And let's put that in parentheses because again, I don't want to make it an official step. Because again, when I say 4th step, what I'm really talking about is the relaxation of the muscle, or if you think about it, what we're really talking about is all the termination processes. right? That's really what we're putting at. So the good news is this last step that we're going to talk about, there's no new information. It is all stuff we have already talked about before. All right, so the good news is there isn't any new information. We're just basically putting question 2, question 5, and like we said, contractile cycle is so simple to end. Basically, when you terminate excitation contraction coupling, you terminate the contractile cycle. So really, it's basically just putting 2 and 5 together. You put 2 and 5 together, and that equals 7. So it really isn't a new essay question. It's just taking this and putting them together. And if you put them together, we get relaxation of the muscle. So let's go ahead and take a break. We will take a 10-minute break, come back at 2:30. And I'll restart the recording then. We will go over this last step, which is the contractile cycle. And then from there, we will dive back into our gross muscle anatomy. So what I'd want you to do during this break, if you haven't done it already, grab your red pens and your blue pens, grab your study guide and your bones picture so that we can start going over those things together, and we'll talk about that as well. All righty, see you in 10 minutes. All righty, we are back. So our goal now for the last thing we have to do from a lecture standpoint is to think in terms of how we relax the muscle. All right, once we get the muscle to contract, we need to relax that muscle. And I'll feed you baby birds step by step, but you guys got to give me the very beginning. How does this process start? If we contract a muscle and then decide we want to relax it, what is the very first thing we have to do? And I'll wait till someone gives it to me. What's the first thing we have to do? Contract your hand into a fist right now. Squeeze it hard. There you go. That's the magic word I was looking for. Absolutely. The very first thing we have to do is make the decision. Again, I cannot emphasize it enough. When you contract and relax your muscles, it is a conscious, voluntary act. So the very first thing you have to do is you have to make the decision to relax the muscle. That is the very first thing we're going to do. As we said when we were talking about termination of the communication at the neuromuscular junction, when we make the decision to relax the muscle, we stop producing neural action potentials. And so, of course, as that means, there are no more big positive signals going to the synaptic end bulb. Perfect. If there are no more big positive signals going to the synaptic end bulb, then that means the immediate effect of that is our synaptic end bulb repolarizes. Of course, that means that the synaptic end bulb repolarizes, which again means go back to rest. And what's the immediate effect of that synaptic end bulb repolarizing? What was making wonky things happen in our synaptic end bulb?
Calcium.
Calcium, absolutely. So what's going to happen is when our synaptic end bulb repolarizes, that closes the calcium channels. So no new calcium into the synaptic end bulb. All right, excellent. Of course, we still have an issue of there being calcium in the synaptic end bulb. So calcium is pumped out of the synaptic end bulb. And when the calcium is pumped out, no more exocytosis of acetylcholine. All right, so no new acetylcholine into the synaptic cleft. Excellent. We are not adding any more acetylcholine in the synaptic cleft, but just like calcium, we have to get rid of the old acetylcholine that's already there. So we have to get rid of the acetylcholine in the synaptic cleft. And anyone remember how many ways there was to do that? Just have to guess. There you go. Three ways. There's one primary way, but there were actually three total ways, right? Way one was just diffusion, right? Diffusion away from the synapse. Way two was endocytosis, right? So basically some of the acetylcholine gets taken back up into the synaptic end bulb. So endocytosis into the synaptic end bulb. But, and you're kind of right about this, the main way, and this is indeed the main way, is that enzyme, acetylcholinesterase, which again, you can then abbreviate ACHE after you've written it out once. That, oops, there. Enzyme acetylcholinesterase breaks the ACH down. Excellent. With no acetylcholine in the synaptic cleft, that means that our chemically gated cation channels are going to close. When they close, no new sodium into the muscle cell. Yes, it also means no new potassium out. But remember, it was sodium that was depolarizing our muscle cell. And so with no new sodium entering into our muscle cell repolarizes. And when our muscle cell repolarizes, there is no new muscle action potentials at the motor end plate. And if there's no new action potentials, muscle action potentials at the motor end plate, there's no muscle action potentials to spread down the sarcolemma. That means no muscle action potentials down the transverse tubules. And if that happens, then our triad repolarizes. If our triad repolarizes, our voltage gated calcium channels on the sarcoplasmic reticulum close, and there is no new calcium into the cytosol of the muscle cell. Once again, we still have the same issue we had before. We have to get rid of the calcium that is already there. And luckily we have pumps that move the calcium back into the sarcoplasmic reticulum. With no calcium, troponin goes back to its original shape. Troponin rotates the tropomyosin back over the active site. Once it's back over the active site, myosin can no longer form a cross bridge and if we can't form a cross bridge, we cannot produce tension. Our muscle relaxes. Notice, as I said at the very beginning of this section, these are the longest processes, but if you think about it, every single individual step is not particularly hard. And as intimidating as this looks when you look at it on the screen, there is literally no information on here. Everything on here are things that we have already talked about. There is nothing new on the screen here. All right. The last point I want to make is, as I mentioned when we were talking about the 7 possible essay questions, this really is a combination of the termination of communication at the neuromuscular junction and termination of excitation contraction coupling. And it's important to remember that as well. This is a continuous process, but we've broken it up into more easily digestible pieces. So if you think of it in terms of termination of communication at the neuromuscular junction, basically that's here from when we make the decision to the point where no new muscle action potentials are produced at the motor end plate, and no muscle action potential spread down the sarcolemma. Right? If we think of this in terms of termination of excitation, contraction, coupling, then if you think about it, because remember the end of one step is the beginning of the next, it's going to start with no new muscle action potential at the motor end plate, no muscle action potential spreading down the sarcolemma, and it's going to go to the point where our myosin can no longer form a cross bridge because the troponin rotates the tropomyosin back over the active site. Yes, technically it goes to there. So if I ask any of those 2 individual questions, and now you can see why we break it up into more easily digestible pieces, because this part in red or this part in blue is a little less intimidating. And then of course, hold on. If we just wanted to talk about termination of the contractile cycle, that's it, right? Again, for consistency, so we have all 3 processes here. All right, termination of contractile cycle is basically 2 sentences. Troponin rotates tropomyosin back over the active site, and the myosin can no longer form a cross bridge. So you can see why that doesn't make a good essay question. But technically, that is our 3 chunks, our 3 processes, and how we terminate. All righty, questions on that? All right, excellent. With that, that is everything we have to talk about from a lecture standpoint today. So I'm going to go ahead and clear this drawing. And what we need to do, let me stop sharing this for a second. Oops, hold on. Excellent. And then what we can do now, I need to close that. Oops, hold on. All righty. Oops. I can find my muscle list. So let's start by coming back to our lecture again. So we are there. We left off last time and we were talking about the muscles of the head that we needed to know the origins and the insertions of. We did talk about the occipital frontalis because that is an important one, and again has two bellies, so it has that magical B specific that you will see when I ask you about that on the exam. The only other muscle that I really wanted to talk about, because when you look at a lot of the pictures and the illustrations and the models in the classroom, although we're not going to be able to play with the models in the classroom as much, so it isn't as big of a deal, is this one down here, the platysma. The platysma is, you can see in this nice illustration from your textbook, is a big, huge, superficial muscle that sits over the top of all the others. So notice like the sternocleidomastoid, it goes over the top of the masseter. And so basically it drapes over the lower jaw and down into the neck region. Right. And so again, when you look at a lot of the models we have in the classroom, like for instance, if we, let's go ahead and do that. See what we have there. If we go back to, excellent. If we go back to the visual anatomy lab, we look at the muscular system, we look at the torso, and we see the head models. Notice here, it's not on this model so that you can see all of the muscles in the neck. Again, same model, so we can't see it here, but where's the head? Where's the other head? We had the other head in here somewhere. Hold on. Huh. All right, well, let's cheat then. If we look at the respiratory models, I know have it because I was looking at it from there. So again, I'm just trying to make a quick point. There's our half head chart. We know that well. You don't care about any of these things, but I swear the head was on here someplace. It's so weird. All right. Now I'm confused. I don't remember where I saw it. I guess it doesn't matter. The point is, most of the models and stuff that you have aren't going to show it. So let's go back here. However, illustrations like this will show that platysma. So depending on which you're using to study it, you won't see it. Oh, I know where we saw it. It was on the, it was in the personal anatomy lab. That's where it is. Let's go back here and instead go to your chapter guide. If we go back to our home of our study area, under the lab tools, under the practice anatomy lab, under models, and when we look at muscular system and we look at the head, There it is. So that's the torso model we have. There's the half head that we have. Notice the half head model we have, again, down here, basically draping up over the top of the jaw and draping over the surface of the neck would be the platysma. It's a big, huge drape-like muscle that's over there. And so it's not on any of our models, but I guess if we end up having this as a lab exam online, then you will be able to use illustrations like the ones from your textbook, and there you can very clearly and nicely see the platysma. The muscle chart that we have in the classroom shows it as well, so that's usually the point that I try to emphasize there, because there aren't a lot of places to see it. But I guess the nice thing about if we're doing this online is that we will have those types of opportunities. All right. What we want to switch to now instead is to talk about the muscles of the torso, the muscles associated with the the muscles associated with the moving of the torso. The first one at the top of our list is the trapezius. Notice when we look at the trapezius, you can see where it gets its name because it has a big, huge, broad origin. It has, where's my annotation tool? Right, so I'm gonna cheat and use my highlighter. A big, huge, broad origin that comes down here, but I went a little too far. And then basically kind of a smaller origin there. And then it has two converging portions to the muscles. So basically it forms a trapezoid. So we have that trapezoid shape to this muscle that we see there. Notice when you look at the origin, and this is where we can go to our bone picture. When we look at our bone picture, and we need to slide this over here. I have too many toolbars. There we go. And we look, oh, where's my trapezius? Oh, the next one, isn't it? There we go. Here's the trapezius up here. Perfect, we found it finally. When we look at the origin, and remember origin, we're going to draw in red. And I'm going to use that highlighter thing again because I like that. Notice its origin is all of the spinous processes starting from C7 all the way down. Right, so as we go to all the way down, from C7 all the way through all of the thoracic vertebrae through 12. So we have those there. Notice also it originates from up here on the occipital protuberance. Where it doesn't directly attach is to the spinous processes of C1 through C6, partially because C1, the atlas, doesn't have a spinous process. But the other reason it doesn't have an attachment there is because it would limit the movement of the neck. And we don't want that. So it doesn't actually attach directly to the bones in this area. Instead, in this area, it actually attaches to, and I'll just do this kind of as a dotted line, that big elastic ligament that we talked about, which is in the back of the neck region, that ligamentum nocha. So basically, we have a continuous attachment, bone to bone, but there is a small portion of it that attaches to the ligament, that elastic ligament, that ligamentum nocha, instead of directly to the bones. And that allows that way we get this long continuous origin, but it doesn't restrict the movement of our head. It doesn't restrict the movement of our neck. All right, that is the origin. What we need to do now is worry about the insertion. Notice when we look at the trapezius, it is a big, superficial, mostly posterior muscle. Clearly, its origin is all posterior. But if we look closely at its insertion, we see that it isn't. part of its insertion is along the spine of the scapula, out onto the acromion of the scapula. But notice the other place where it inserts is out here on the lateral third of the clavicle. And if you think of the clavicle and where it is located, it is actually an anterior bone, right? If you feel your collarbone, you can feel that clavicle. And so it's out there on the anterior third. We don't think of it as being a partially anterior muscle too much unless you have that situation that we've talked about where those bodybuilders. In those bodybuilders, these muscles can get incredibly large. And notice this big, huge mass of meat that we see up here on the top is that little bit of the trapezius that is coming onto the anterior side of the body. So we have this big, huge, almost triangular shaped muscle on the back with a little bit of it sneaking onto the front. And that is our trapezius. All right, so we can see that really nicely when we look at it there because it's coming out anteriorly out onto this bone in this location. All right, so that is the origin and the insertion of that. That can be a little bit tricky. Notice we go back here, and unfortunately my drawing is gone now, but if you think about it in the way that it is, I'll just simplify it this time, draw it down this way like that for our origin. And our insertion is out here. Notice the joint that it crosses, or really the bone that it is moving the scapula. Notice it doesn't attach to the humerus. It doesn't move the arm. What it moves is the shoulder. And how many actions does the trapezius have? If only I had a list of them in front of me and I could count. There you go, excellent, 4 of them, perfect, right? 3 of them are the big R's, right? Back in ancient times, by ancient times, I mean when I was in school, when you talked about the 3 R's in elementary school, it was reading, writing, and arithmetic. And if you think about it, 2 of those 3 things aren't really R's. So it tells you something about the education I got growing up in elementary school. Kids nowadays, my youngers, when they're in school, the 3 R's were, of course, reduce, reuse, and recycle. That was all the big rage now. Well, here, our 3 R's of the trapezius are going to be to raise, to elevate the shoulder. So elevate, raise. To retract, pull it back. Because if you think about, so again, it's going to pull it up. It's going to pull it back. So those are going to be the movements. Sorry, I apologize for that. Perfect. Pull it up, pull it back. The other thing that we're able to do is we are able to rotate it. If you think about as you are abducting your arm, so think about abducting your arm as you bring your arm up. Notice at first as you're abducting your arm, you're swinging your humerus out until your humerus is about parallel with the glenoid cavity. If you continue to elevate it without moving the scapula, the neck of the humerus would bump against the acromion and the clavicle, and it could damage it. So what happens is, as you continue to abduct your arm all the way up, what's going to happen is your scapula is going to rotate. So the bottom part rotates this way, and the top part rotates that way. So we have our 3 motions: an elevation, a retraction, and a rotation of the scapula. So those are the 3 R's of the scapula, the 3 R's of the trapezius. Now, there's one more action as well. Remember, as we've seen, and let's cheat now and go back to the picture. Not that one. This one. Again, we can see how it is grabbing on to the spine and the acromion, fascicles coming forward to the anterior side on the clavicle, and so it can pull on the scapula that way. But notice if we just contract these fascicles that are up here on the neck, and we use other muscles to hold our shoulder in place, instead of our shoulder coming up, what will actually happen is the back of our head will come down. And of course, that action of the back of the head coming down is an extension of the head. So if you look up at your ceiling, you are extending your head. So 4 actions. We are going to raise the scapula. We are going to rotate the scapula. I'm sorry, retract the scapula. We are going to rotate the scapula. And we are going to, if we just use the superior fascicles, we are going to extend the head. So those are the 4 actions of the trapezius. All right, so we've identified the origin, we've identified the insertion, we have identified the 4 actions. So we have that for our trapezius. All right, questions on that? Excellent. All right. The next thing we want to look at is the serratus anterior. The serratus anterior is kind of interesting in its location. As its name indicates, anterior, it is on the front of the ribs. And notice as we look at it, and I'm going to stick with my highlighter, I like that. And we'll highlight it in something light colored this time. Purple, there we go, excellent. Notice it has, oh no, it doesn't show. Oh, darker better? No, that stinks too. Black? There you go. Notice that it has all of these attachments on the anterior part of the ribs. So notice if you look at the list and what its origins are, it is the origins starting up here with 1, 2, 3, 4, 5, 6, 7, 8. It attaches to the front of all of the ribs. That is hopefully easy to see. Right? Notice here, they've been nice enough to remove a big, huge pectoralis major and a pectoralis minor and some other muscles so we can see that. But notice when those muscles are in place, we can still see a little of them peeking out from underneath. And again, this isn't just something we can see on an illustration like this. Here's our bodybuilder. And notice here, we can actually see those little fingers of the serratus anterior attaching underneath the pectoralis major. So we can see those on our bodybuilder as well. That part of our serratus anterior is pretty simple and straightforward. However, it does get a little tricky, and for this I want to actually draw this. So let's go to our whiteboard. Let's clear our whiteboard off. And let's take a look at the rib cage. Cheat. Oops. Oops, if I make it a color we can see. Excellent. Let's pretend that is our rib cage. All right. Here is our vertebrae on the back. Here is our sternum in the front. And most importantly, here is the scapulus, our shoulder blades. Now, we just finished pointing out that our origins are going to be on the front of the rib. So here is an origin on this side. Here is an origin on this side. They're on the front of the ribs. If you look at the list, where the insertion is located is on that scapula, but notice it's on the anterior and medial border. So it is on the anterior and medial border of the scapula. What this means is the serratus anterior, and let's pick a color, green. Our serratus anterior starts on the front, wraps around the ribs, goes underneath the scapula, and then connects to the medial border of the scapula. So again, we see it in the front, it wraps around the ribs, and attaches to the medial border of our scapula. It wraps around it in that way. We see kind of what that looks like when we look at the, again, picture of it. Here we see them wrapping around. Notice here's the scapula. We can see it going under the scapula. Notice that they peel more of the muscles away. Again, we can see it going under the scapula, and we can actually see where it's attaching to that medial border of the scapula from the back. So here we can see that scapula from the back, and we can see it's going around onto the medial border. So it does do a nice job of kind of showing that. If we go back to our drawing of it, though, the advantage of the drawing is it helps us to understand our actions. So when we think of the actions, its job is actually to hold the scapula stable against our ribs. Basically, it pulls the scapula forward, and that stabilizes the shoulder. Now, of course, pulling the scapula forward is, of course, what we call protraction. However, if you notice, it really can't pull them forward because those pesky ribs are on the way. If you reach out towards your computer screen, and don't touch it unless it's a touch screen, but as you reach towards you forward, what you see as you feel your arm going forward, you feel your scapula actually moving, around the edge of the ribs. So it does pull it forward. If you were to hug yourself, for instance, you could feel the scapula is coming forward, but also what they do is they slide around the edge. So while what we call it a protraction, the other thing we can think of it as doing is to be pulling the scapulas away from the midline. So we could also call that an abduction. Abduction. So both of those are acceptable. You can refer to its action as a protraction or as an abduction because it's basically the same thing. It is pulling it forward, but because those pesky ribs are in the way, it has to pull it around the side. So it is an abduction. Notice also, as it's doing this, the other thing that it does is, if you think about it, the other thing it's going to do is it's going to squeeze the thoracic cavity. When you take that forced exhalation, blowing out your candles on the birthday cake, one of the muscles that helps with this is the Serratus Anterior. This Serratus Anterior actually squeezes the thoracic cavity, and it is one of the accessory muscles we use in breathing. So it's going to be an important breathing muscle as well, because basically by pulling it forward, we're squeezing the thoracic cavity so that we can force air out. All right. Questions on that? Notice, and I'm going to actually stay here first before we leave. I have to change my drawing, make it a little bit fancier. Where if we're going to make this let's stick with black. If we are going to make this the vertebrae, I need to give my transverse processes and my spinous process for all of that to my vertebrae. Put a big V in here to remind us that this is the vertebrae. Because if we look at the next muscle that is on our list, the next muscle is the rhomboids. This is a muscle group. And again, the muscle groups are groups of muscles, in this case, the rhomboids major and minor, but we don't have to worry about that. Notice the origin of the rhomboid is out here on the spinous process. Oops, it's supposed to be red. All right, undo that. Draw a red circle. The origin is out here on our spinous processes of our vertebrae. Not all of them, notice, just basically C7 through T5. And we'll draw that on the pretty picture that we have in just a minute. But I want us to see this relationship to the scapula. Because where our rhomboid inserts is onto the posterior medial border of the scapula. So notice this muscle basically connects the scapula to the center of your back. So like I said, you see your long lost friend that you haven't seen forever. Again, assuming the coronavirus isn't around, you go and you give them a nice big huge hug. You're going to use your Serratus Anterior for that. However, then you realize the reason you haven't seen them in 2 years is because they owe you $150. So you bring your arm back to throw them a punch, and as you bring that arm back to throw them a punch, you are using this to pull your scapula back. In fact, if you or make a rowing motion where you pull both arms back like you're going to punch at the same time, which would be a silly thing to do, but if you do that, what you feel is you feel this scapula being pulled back that way, this scapula being pulled back that way, and you can actually feel the muscles bunching up in your back as you're pulling on those rhomboids. So you can actually feel that as it is occurring. So let's take a look at that here first. So notice here are the rhomboids, again, the minor and major, but you don't have to worry about that. So we just think of them as a single muscle group. Attaching to the spinous processes of C7 down through T5. And here they're inserting onto the medial border of the scapula, pulling the scapula back. So Serratus Anterior pulls the scapula forward. This one pulls the scapula back. All righty. Questions on that? And notice, again, as it pulls it back, it brings it towards the midline, so we can think of it as a retraction. We can think of it as an adduction. Both are acceptable terms. All right, the good news is we only have one more muscle we need to know the origin and the insertion for, and that is this one here in the trunk region, and that is the pectoralis minor. The pectoralis minor, as we can see here, it originates from the ribs of rib 3. Oops, where's my annotation? There it is. Anterior surface of ribs 3, 4, and 5. This is its origin. And notice it inserts into the coracoid process. As we've talked about, the job of muscles is to bring the insertion down towards the origin, and so that's what it is going to do. We're going to get that downward pull of this. Because it's attaching, again, not to the arm, but to the shoulder, that's why it's considered a trunk, it brings the scapula down, so it depresses it, and it brings it forward, protracts, or remember, we think of it as abducts. So either way is acceptable. It brings it forward and it brings it down. But again, we have the ability to be able to stabilize the shoulder with other muscles so that when we contract it, what happens is instead it pulls the ribs up. And so that would be, again, one of those inhalation muscles that help us make that big inhalation. So if you look, there are 3 actions to the pectoralis minor. They are to depress the scapula, protract the scapula, bring it forward, or elevate the rib cage. And this is another muscle that helps us to breathe. All right, questions on that? All right, the last thing that I wanted to do for today, actually the last 2 things, is first talk about this muscle, the diaphragm. Again, we don't need to know the origin and insertion of it, but we know it is, because on the very first day of class, we talked about how it is that anatomical boundary between the thoracic cavity and the abdominal pelvic cavity. Here we see it from an inferior view. It's a big, curved, bell-shaped muscle that forms that mechanical barrier. So notice things like your esophagus, so your food can get to your stomach, your blood through your aorta, your blood through your inferior vena cava. They have to have tiny little holes in this muscle that help to allow the blood or the food through that barrier. And that's what this is. The diaphragm is basically a supportive barrier. It is a bell-shaped muscle, which means it is curved. And when a curved muscle contracts, basically what happens, all muscles get shorter. So when it gets shorter, it flattens out. When it flattens out, that increases the space of the thoracic cavity. Now the volume in the thoracic cavity goes up. And of course, as we know, if volume goes up, pressure goes down. and you breathe in. So basically the diaphragm contracts, providing that support, but when it contracts, it is what allows you to take your inhalation. While you're sitting here calmly in the classroom right now, well, not in the classroom, but sitting here calmly in class, you're pretty much just using your diaphragm. If you look down, you shouldn't see your chest heaving as you breathe, unless you've been doing this on the treadmill while you've been listening to all of this, which I guess isn't a horrible idea. But if you're just sitting there like I am and you look down, your chest should not be heaving, because it is primarily this diaphragm that is doing the contraction for us. However, it turns out the diaphragm is not the only supportive structure. We have another supportive structure, and we'll get to these in a second. This is the one I want. We actually have what we also call the urogenital diaphragm. The urogenital diaphragm is the support structure for your viscera in your pelvis. We talked about the pelvis. Here we see the ischial tuberosities of the bones. We have the muscles that support the structures like the anus and the vagina in the female, and males have it too, supporting the anus and the penis in this area. This urogenital diaphragm's job is to provide structure and support. You elevate it or contract these muscles to facilitate defecation or to help in urination or stopping in urination. The other thing, let's go back to the females, because this is one of the things where it'll be more relevant. The other issue is that females get the honor and the privilege of giving birth, right? They basically have to pass a basketball through their vaginal canal. When they do that, that weakens this urogenital diaphragm that provides that support. One of the implications of that is a female, typically shortly after she gives birth, the weakening of this muscle makes it harder for her to contain her urine. So if a female who's just given birth sneezes or coughs, or even sometimes laughs too hard, a little urine can leak out as a result of that. However, in extreme cases, actually the vaginal canal can actually prolapse, where it descends beyond the urogenital diaphragm, and it has to be put back in surgically. All right, a very serious condition when that occurs. Luckily, like all muscles, this can be strengthened. Now, this urogenital diaphragm is formed by many muscles, but primarily it is formed by a muscle group known as the levator ani. The levator ani, and again, we have this in both males and females, is a muscle group made up of two muscles, pubococcygeus and iliococcygeus. And even though those are both incredibly fun to say and spell, I'm not making you know them. You just need to know the muscle group, the levator ani. Right? Now, again, any woman who's been pregnant before probably knows this. There is a special set of exercises they encourage you to do to strengthen this levator ani muscle. Anyone tell me what that is or type it out? If you're too embarrassed, you can write it privately to me. Say again?
I said Kegel exercises.
There you go, Kegel exercises, absolutely. Those Kegel, right? And of course, why are they called Kegel? Because good old Bob Kegel's the one who first described them. Those Kegel exercises basically are where you contract the levator ani muscle. It would be, again, if you've never given birth, if you've never done this before, think of it in terms if you were urinating and you wanted to stop midstream. That contraction you would make of your urogenital diaphragm is that contraction of the levator ani. And so basically what you do is you take that contraction, you hold it for 10 seconds, you relax, and then you do it in sets of 10, right? So you contract and hold it, you contract and hold it, right? I'm sure you're doing a couple of it. I just did 4 of them while we're sitting here. But that contraction and that holding of that for the count of 10, that is that Kegel exercise. And the whole point of that is to provide that support for that levator ani, which is part of that urogenital diaphragm. All right. As I look at the rest of the list, the intercostals are also accessory muscles for breathing. Let's go back to that real quick because there is just, actually, you know what? Those and the abdominal muscles, I do want to actually have more time to talk about, but I want to talk about them together. So I think this is a good stopping point. I think we've had a nice, good, long lecture with a lot of content. So I think this is a good stopping point. So We will save the rest of those, and let's go ahead and, oops, I want to do that. So we can come back to here. Excellent. We'll talk about the intercostals. We will talk about the intercostals. We will talk about the abdominal muscles in our next class. So there's a question on the list study guide. The blank boxes we don't need to know. That is correct. So again, for the origins and the insertions, those ones that have dotted lines does not mean they don't have origins and insertions. They certainly do. It just means you're not going to be responsible for them on the exam. As for the lab and lecture exam, as always, all the material we learned in this class, this is one class. We have different modalities for learning them. So for instance, lab and lecture component of it. And lab exams are better for identification. Lecture exams are better for, descriptions and things like that. But absolutely, you're responsible for all of the material we cover in all the exams. So on the lab exam, I may ask you what the internal, to identify the internal intercostal, but I could also ask you its function. Right? I could ask you processes during the lecture exam, but I could also ask you to identify all the muscles that play a role in respiration and to ask you which ones inhale and which ones exhale, right? So absolutely, all the material that we cover, you're responsible for on both the lab and the lecture exams. Great question. All right, any others? That is, like I said, more than enough information. We've covered a tremendous amount. The good news is you have 5 whole days to decompress from this lecture and to make this lecture make sense. When we come back on Tuesday, one of the things I want to do is sit down and go through the entire process of a muscle contraction, make sure we have it. And it would be nice if I would occasionally get, more than occasionally, if we would get some interaction with this. Again, the way you show me you guys are knowing this thing is by being able to answer the questions. When you have the list in front of you, when you have these things in front of you, these are the easy times to do it. So this is your opportunity to show me that that this is working, that this time and effort that I'm putting in. You got to remember, I'm not getting the feedback from you that we would normally get in the class. In the class, I can see, right, that whether you're asleep or not, or whether you're paying attention, whether you're furiously scribbling, whether your hand hurts when I stop talking, right? I get that feedback that you guys are active in this. You guys got to try to give me something so that it helps me to keep going with this. So I appreciate those that are speaking up and would love if more. And again, if you don't want to use your microphone, don't want to speak, then just type it in the chat box. That'd be really great as well. But it helps me a lot, so I would appreciate that. But like I said, we covered a ton of stuff, so we'll do a review at the beginning of next class. And then now that we finally know how a muscle contracts, we can start building up. We can go from the proteins that we've looked at to a sarcomere, from the sarcomere to the myofibril, from the myofibril to a muscle cell. And then we can start talking about where the energy comes from and fatigue and all the fun things and the types of movements that we're going to allow. So things get fun from here. So all the long, big, exhausting processes are done, and now we get to the fun stuff. All right. And also during this time, do not lax off on the lab stuff. I know we're not having as much time to work on this together, but it is important. I guarantee you. So let's start here. So as I've mentioned, everything is still up in the air. No one still can give us any certain information of what's going on, but it is looking more and more like this may continue past spring break. What I know is that there's a department meeting tomorrow where we're meeting online and talking about these things, and I hope to, by the end of this weekend, starting next week, have a better idea of what our long-term plan is. Again, you just got your test back on Tuesday. I was struggling to get your test made. I was struggling to get your tests made. Then I had to grade the other classes' tests that they did in person. Then I had to grade your online ones. I've been playing from behind up to this point. This weekend is going to be my first big opportunity to start looking forward and figure out what our long-term plans are, including the exams. What I will tell you is whether this is a station-based exam in the classroom or whether it's going to be online, I guarantee you there will be pictures of bones. And I will point at bone features of bones. And you will need to be able to identify the muscle that attaches to those points. You need to know your muscles on your list. Be able to identify the muscles any way that I show them to you. Models, charts, pictures of models, pictures of charts, illustrations from your textbook, bodybuilders, All of those are things that you're going to need to be able to see and identify on those things. Know their actions, and the bold ones, the ones where they're listed, know the origins and insertions. Normally, this class is very simple and very straightforward. The more time we spend on something, the more important it is. Because of the format, it is harder for me to lead you through the muscles. So we're not spending as much time in lecture or in lab talking about it, but that doesn't mean it isn't going to be important. If we're back in the classroom, this is another one of those 80, 85 point lab exams. If we have to do it online, it's going to be bigger than the last one. My guess is it would still be somewhere between 60 and 80 because I have more time to make it and make it right this time because we're not trying to do it in 2 days before we have to come back. So make sure you're spending time on this stuff. I know the processes are intimidating. You want to spend a lot of time working on those, but make sure you're also doing the lab stuff as well. Make sure you're also doing the anatomy stuff as well. Use those resources. Use the, again, I can't emphasize enough how awesome, where is it? Did I close it? No, here it is. I cannot emphasize enough how awesome your practice anatomy lab thing is. Take advantage of this practice anatomy lab, right, for the muscles, for the, so if we're looking at the trunk muscles, right? Oh, gosh, models, muscular system, right? Upper limb. These are great for helping you to be able to identify. Because again, these are likely to be the types of pictures I would use on the exam. And the best thing about it is these models are the models that are in your classroom. So if we have to do it on the classroom, then this you're going to be able to tell is the subscapularis because you looked at it. And this hiding over here is the deltoid. And this is the bicep brachii and so on and so forth. So yes, so if we have to continue this class online moving forward, then yes, the weight of the online tests will have to come up. I was hoping that this one skeletal one would be an aberration, and so I didn't want it to weigh as heavily on your exam because of the different format. However, if this is our format moving forward, then yes, there's two things that are going to happen. First, the tests will be bigger with more content on them, so they'll absolutely be worth more points. But also, we're also going to have to be more vigorous in the testing process as well. Again, I will have more answers for you next week, and our test is until after spring break, so we'll have a couple weeks to work on that. But if we have to do this online, part of that doing it online is going to involve you using the full Proctorio suite, which means that you will need to have your camera on so that it can monitor you and record you while you're taking the exam. So one of the things that we'll do when we come back is if that's what we're going to do, and again, that's a lot of what tomorrow's department meeting is on, so I'll have more information next week, I will again set up a practice exam so that we can work out all the kinks in that process so that when it comes time to test time, we can just focus on the content and not trying to figure out what the heck we're doing. So that is going to be the game plan. But like I said, I've got this weekend to work on it, and we've got a meeting tomorrow that I'm suspicious may continue on into the weekend as well. But ultimately, I don't know what the plans are. And so I will hope we'll have more on this in there. But yes, if we are stuck online for the rest of the semester, then the tests will be bigger. The tests will be more vigorous because there's a lot of content that we have to cover. All right. Great question. Any others? All right, so at this point, I'm going to go ahead and stop the recording. As always, I will hang around for a little bit if there's any more other questions. Again, if you have good questions, now's the time to ask them so that other people can benefit from them. But if you have more personal questions or anything else that you want to discuss with me, I will hang out for a few minutes to give people the opportunity to ask that, and then I'll close the classroom because once I close the classroom, that is when the recording starts to format. We've had a nice long 3.5-hour lecture, so it'll take a little bit of time to format, but I will get it up onto YouTube tonight if it's not up by this evening or tomorrow morning at the latest, because yesterday's took a long time. But if it's not up by tomorrow, then definitely contact me because there's probably an error, but I'll be watching it as well. All right, any more questions? All right, you guys, I know this is a lot of material, but like I said, you have 5 days to unpack it. Work on this stuff. The point that I want to emphasize when we look at these processes is the processes are not hard, they're just long, right? Yes, it's 15 steps or whatever, however many you want to count it that way, but every single individual step is simple. It is straightforward. So really, it's just putting about all those steps together. All righty. Will there be live models used on the muscular test other than the head that you've used so far? I'm not sure what you mean by live models. You mean models like the one that we have on the screen right here?
So you know how you have that picture of the head? That's like an actual model you could like physically touch. We have like models like that.
Well, yeah. I mean, if you think about this picture here is a model that you could actually touch, right? And again, there are different views of it, but this is actually a picture. This is actually a picture of the model from the classroom. You're talking about the ones that are on the CRC, I think. So like if you go to the, I think I closed it. Looks like I closed it. But here, if we go to that visual anatomy lab, the CRC one, and we click on this, muscular system, notice again, it's basically it's the exact same model. It's just a picture of it from the classroom. So yes, those are definitely pictures of the models as well as the illustrations from the textbook, as well as other things like that. I'm always willing to use. Absolutely, because as long as it's a good example, this is a very good example of the deltoid muscle, right? This is a pretty good example of the triceps brachia that we're seeing here. There's a really good view of the brachialis. So absolutely. So there are definitely some good things on this, and I would definitely be willing to use a picture like this on the exam. Now, I wouldn't use it for the medial head of the tricep brachia because all you're seeing is that tiny bit of it right there. But any muscle that we see a large amount of, and if you could orient yourself on this for the body, then yeah, absolutely, I would use something like this. So I would use these as well as the illustrations from your textbook. So don't just memorize the pictures from your textbook. But also don't just memorize these pictures as well. Because again, if I have a novel example or find a novel picture of these things, as long as it is a good, obvious example, then I will definitely use it on the exam. All right, great question. Will you be available by email during spring break? Yes, absolutely. Again, like you, I'm not going anywhere. So, and especially if we're coming back for the exam, well, heck, one way or another, we're coming back for an exam after spring break. So yes, I'll be spending the majority of my spring break writing both your test and my 431 test. Your test is Thursday, their test is Wednesday. So there's two good things about that. The one is that I have spring break from now to spring break to work on those, but also hopefully I can learn from giving them their exam on Wednesday. So hopefully that will limit the number of problems that we have on ours. So hopefully that will go well. But yes, I will definitely absolutely be available. I may even have some quote unquote office hours. Again, remember, you're always welcome to reach out to me by email, not just to respond to me by email, but if you want to meet and talk about your exam or talk about anything else or go over the arm with me, we can do that through Zoom. Remember, I have my own office room now, my private, it's not private, but my office room, which is basically just my office number, 916-484-8992 that you see on that. So I have a room that I can set up whenever we need it so that I can meet if you want to do that as well. So absolutely, I definitely will be around during spring break because I'll be working on your exams, but I'm happy to help you guys with anything that you need during that time.
At virtual labs on Fridays, are you allowed to interact with the teacher in there? Like, is it on Zoom, right?
I believe Jeff is doing it on Zoom. I'm afraid I don't know anything about it. Jeff's the one who's setting that up. My guess is that he is there to answer questions. But like I said, I'll be honest. I don't know how he's running it. So I know he's running it by Zoom. I know it's virtual. I know he told me he had models. My guess is he's not just going to stand there and randomly rant about the models. Not that Jeff's beyond that, but my guess is he's there to answer questions. The only thing I would say is my concern with that is if he gets, 40 people in there, your particular question might not get answered. So that would be my only concern with that.
So you couldn't like say, oh, rotate this so I can see, the anterior side or something like that. I can't do that.
I assume he'd be able to do that. I mean, again, my understanding is he isn't just going to set up a picture, a model under the camera and walk away. My guess is he's going to be there to answer questions. But what I'm telling you is that I don't know how popular it's going to be. And you got to remember too, he's got 430 and 431 students. And unlike 430, where you're all kind of working on things at the same time, everybody teaches 430 differently. My students are doing respiratory system and urinary system. Somebody else's students may be doing reproductive or digestive or endocrine. So he may be showing a stomach to some students, and then somebody else wants to see a lung, and you want to see an arm. So I'm just saying that I definitely think that you should take advantage of it. Jeff is a great resource, and the fact that he's willing to do this is awesome. I don't know the format by what he's doing. And then my only other concern, like I said, is depending on how popular it is, he may not be able to answer the question that you have. So just be aware of that. So he's a great resource. I encourage you to take advantage of that. You should be taking advantage of all of the resources that are available to you. But I can also address questions and answer questions outside of our normal class time as well. All right, anything else? All right, I'm going to go ahead and stop the recording at this point. If no one else has a question, I'll still be around as people start leaving the classroom in case there's anything else that you want to ask or think about. But otherwise, I'm going to try to end this as soon as possible so I can start getting it to format the video for us. But like I said, I'm here if you have any questions. You guys have a good weekend. Stay safe, stay healthy, and have a good weekend. Study hard.